So in celebration of the recent Nobel Prize awarded for the elucidation of ribosomes, I wanted to make this video about evolution using the ribosomes as one of my examples. Here I will show how molecular interaction leads to enzymatic function and how changes in molecular interaction leads to changes in enzymatic function. This change in the function of enzymes is the essence of evolution. Everything I say here is from this textbook, Leninger, Principles of Biochemistry, so you guys can check it out if you want more details. So first I want to use the example of the How Ribosomes Work video I made a while back. This video is really confusing if you don't actually understand how ribosomes work. So it's a really bad title and I apologize. But you should still watch the video because it uses state-of-the-art technology to simulate the action of ribosomal accommodation of amino acid tRNA. They model every single atom in the complex and use algorithms for natural interaction of atoms. Everything works exactly as it does in real life, and this shows us that we have a really good understanding of this process. And since all the variables and algorithms were of natural means, we know that God isn't involved pushing every single ribosome around. So if you don't understand the basics of ribosomes, please watch the attached videos, which shows you all the basics about what an amino acid tRNA is, what regular tRNA is, what mRNA is, how all of these interact. And then watch the How Ribosomes Work video for some really cool stuff. But it's not just really cool, it gives you an example of how evolution works. So you see, in that video, there's a lot of molecular interaction going on, and that's the theme of this video. The tRNA has to interact with the mRNA so that the correct codons match and hydrogen bonding can occur. And the tRNA has to then navigate through the gates of the ribosome itself. The top of the tRNA has to navigate through the gates while the bottom is attached to the mRNA. So in order to navigate through the gates, it needs to interact with the A loop and the H89 sequences and has to go through that corridor and go to the center of the ribosome. The tRNA does. If the wrong tRNA is attached to the mRNA, in other words, it's the wrong codon, then there will be a weaker interaction with the mRNA. The hydrogen bonding would be weaker, which means it would be much harder for the tRNA to go through this passage, to go through this corridor in order to get to the center of the ribosome. Now, let's say that a random mutation causes the H89 to get bigger, which means that the corridor now gets narrower because H89 is one of the gates of the ribosome. Right? So the corridor is now narrower, and the tRNA has to squeeze through the space with even more effort than it had to before. This means that it has to be even more tightly bound to the mRNA than it had to before. And this means that the wrong tRNA is not as likely to bind because the hydrogen bonding is weaker. And so the right tRNA usually binds, and the resulting protein will be very faithful to the mRNA. Now let's say that a mutation makes the H89 smaller. In other words, the corridor gets bigger. This means that the wrong tRNA is bind to the mRNA and get through that corridor because less effort is required than before to get through that corridor. So a weaker interaction with the mRNA is able to do it. So this means that the end product will have lots of mistakes. Do you see how molecular interaction leads to function? This is really important. But more important is that the change in the molecular interaction of the tRNA with the gates changes the function of the ribosome. In other words, the end product is either more accurate or less accurate. The end protein product is either more accurate or less accurate. So now let's see how scientists use this idea in a practical method called Selex, which stands for Systematic Evolution of Ligands by Exponential Enrichment. This is essentially evolution speeded up, and it takes advantage of this molecular interaction. Let's say that you want to produce an RNA molecule that binds to ATP from scratch. I'll put this illustration up in the description box to make it easier for you to follow. But what you do is you start off with a column, right, a plastic column, with resin attached around it, and there's ATP attached to the resin. And what you do is you send 10 to the 15th power of random RNA sequences that are 25 nucleotides long through the tube containing the resin. This high number, the 10 to the 15th, ensures that there's a complete randomization of 25 nucleotides. So this is as random as you can get. So what happens is there are certain RNA sequences that will weakly interact with the ATP and stick to the ATP, and there are others that will flow right through the column and they'll get washed away. So what you then do is you take those RNA molecules that bound to the ATP and put them through this cycle where you convert them from RNA to DNA using reverse transcriptase, and then you convert them from DNA back to RNA using RNA polymerase. RNA polymerase is what we use in our bodies, in human beings and every other living creature, to make RNA from DNA. So this is a natural process. So anyway, the RNA polymerase adds new random mutations to these sequences. And what you do with these new sequences, with the new mutations, random mutations, is you run them again through these through that column, and then again you collect whatever sticks, you run it through the RNA polymerase, and then you run it through the column again and again, a dozen times, until at the end what you have is RNA sequences that bind extremely tightly to ATP. So once again, interaction leads to function, and changes in those interaction, in other words a random mutation, leads to a different function, in other words a more tight binding of the ATP. Using this process, Selex, they have isolated artificial ribosomes that synthesize artificial peptides. What is interesting is that these artificial ribosomes have highly conserved sequences found in all peptidyl-transferase active sites in the normal natural ribosomes we have in all cells. So basically, even though we started out with complete randomization of RNA molecules, in the end, you end up with a ribosome sequence which is exactly the same as in nature. This shows us that it's not the randomness that's what's significant here. It's the natural selection. Since natural selection is same in both cases, in both the artificial ribosome and in the natural case, it forces the randomness to go to a certain conserved sequence, and that's what's important. The randomness is important, but the fact that everything seems designed, the fact that they converge onto the sequence, shows that the selection is also important. So they've also used a similar process to determine the complicated pathways that are required for many cellular changes. So for example, we have 20 amino acids that are commonly used in our bodies. So how would nature add a new amino acids? How do we get from 19 to 20 or 20 to 21? In order to do this, we need three things: a new amino acid tRNA synthase, new tRNA, which proper codon, and a new amino acid. So for those of you who don't know what the tRNA synthase is, what it does is it loads the amino acid onto the proper tRNA. So the tRNA synthase has to be unique, along with the tRNA with the proper codon and the new unique amino acid. So this is a complicated process. Yet by running many cycles of positive and negative selections on tRNA synthase and tRNA, they produce new interaction and new function. They have produced new tRNA synthase along with the new T codon, and they had a new amino acid, which they had from scratch. So there are two evidence that you can use against. There are two arguments that you can use against this evidence. First is that this is an experiment done in which you need living animals necessary. In other words, you need to produce that RNA. So you need living creatures from start. So I'll get back to that. This is abiogenesis. So I'll get to that later. But what I want to discuss now is what people say. What people would argue is that this shows that intelligence was necessary because scientists were doing it. Well, consider what scientists did that was so profound and unnatural. They ran RNA through a column that interacts with molecules in that column. The selection is natural, the production of RNA is natural, then the reproduction is natural, the random mutations are natural. All the scientists did was run that RNA through that column. In other words, they put that RNA really close to that ATP. And nature can do this too. Nature can bring two molecules really close together by a process of diffusion and other methods. So but again, this takes a really long time in nature for them to bring molecules really close together, and that's why evolution takes millions of years in nature.